Hello students, I'm Dr. Divya on behalf of TeenEinstein. So now we are in the second chapter, sexual reproduction in flowering plants. So in this chapter we are learning about a flower, then pre-fertilization, then double fertilization, post-fertilization, then apomixis and polyembryony, then some important definitions related to this sexual reproduction of flowering plants. So we are learning about all these things. So reproduction it's an important process without which species cannot survive, and an individual, how it is increasing its number? By reproduction, by asexual or sexual method of reproduction. So sexual mode of reproduction means that is creation of a new organism or new variants, so that organism will be having better or advantages compared to this parents. So all flowering plants shows sexual reproduction. So that creates lot of diversity, and in this chapter, sexual reproduction of flowering plants, we are learning about the morphology, structure and process, different process of sexual reproduction in flowering plants, because sexual reproduction is a very complex and time consuming process. Okay. So flower, first we have already learned about several concepts of this chapter. So we learned about flower, which is an important structure, which is the site of sexual reproduction. So in most of the occasions we are depending upon flower. Like flowers, they are the best sites of sexual reproduction. They are the sites of sexual reproduction, and the structure of a flower we have already learned in the previous classes. We were discussing about the structure of flower. So a typical angiosperm flower, it is having 4 whorls or floral appendages. They are called calyx, corolla, androecium and gynoecium. In this, calyx means that is sepals, then corolla, that is petal, then androecium is the male reproductive unit and gynoecium is the female reproductive unit. Among this, calyx and corolla are called as non-essential whorls, and this androecium and gynoecium, they are the essential whorls without which sexual reproduction is impossible. Okay. So let's have a recap of this pre-fertilization structures and events. So just much before the actual flower which is seen on a plant, the decision that the plant is going to flower, it has taken place already. Okay, so number of hormonal changes and structural transformation occurs before that flower formation. So that shoot meristem, that apical meristem will transform into a reproductive meristem. Then that reproductive meristem grows to form an inflorescence axis, and over that, or this floral primordia develops, and this primordia that will grow into floral buds, and that buds will develop into a complete flower, which is a reproductive structure. Okay, so in the flower, the androecium and gynoecium, they will develop separately. So stamen, that consists of mainly 2 parts. Okay, anther is the terminal structure which is bilobed, and filament, it is a long stalk, a slender stalk-like structure. Then we have learned about the typical structure of an anther. Anther is bilobed, and that anther lobes are separated by a deep groove that is called as connective, and each anther is a tetragonal structure, and it is having 4 sides that consists of 4 microsporangia, and these microsporangia are located at the corners. Okay, so in each lobe there will be 2 microsporangia. So the mature anther is having 4 microsporangia. So microsporangia forms pollen sacs, and on maturity this pollen sac, it will open and it will release pollen grains. Okay, and the structure of microsporangium we have learnt. So in a transverse section, a typical microsporangium, it appears circular, almost circular. It's not exact circular, it's almost in circular structure. So primary sporogenous cells, these primary sporogenous cells form microspore mother cell. These microspore mother cells are diploid. All this we have discussed. And anther is having wall layers. It consists of epidermis which is the outermost single layer which is having protective function. Then that epidermis, after that endothecium, cells over this layer, it is having alpha cellulosic fiber bands. Okay, and next is middle layer. Cells of this layer, middle layer, that is ephemeral, and it is around 1 to 3 layers. Okay, and when the anther matures, this layer that degenerates. Then next is tapetum. This is the innermost layer. Tapetum is the innermost layer, and this surrounds the sporogenous tissue. Okay, so the sporogenous tissue means when this anther is young, a group of compactly arranged homogeneous cells, these are mostly parenchymatous cells, and this tissue that occurs at the center of each microsporangia. These are mostly parenchymatous tissue, and they are compactly arranged tissues which are homogeneous in nature. That is called as sporogenous tissue. Then microsporogenesis. Then the formation of this haploid microspore from a diploid. From where it will form? It will form from a diploid microspore mother cell inside pollen sac. And which division is taking place here? Meiotic division, that is called as microsporogenesis. Okay, so formation of haploid microspore from microspore mother cell which is diploid, that is called as microsporogenesis. And this haploid microspore formed from a single microspore mother cell or pollen mother cell. They are arranged in the form of 4 cells called microspore tetrads. Okay, they are called as microspore tetrads. And next we learned about pollen grain, the structure of pollen grain. So the pollen grain, that is representing the male gametophyte. So these are spherical structures, pollen grains, and it is having cell wall, and this cell wall of that pollen grain is called as sporoderm, and it is having 2 layers, exine and intine. Exine is the hard layer, which is outer layer, which is made up of sporopollenin, that is the most resistant organic material ever known, and that can withstand high temperature, strong acid, alkali, etc. And next is intine. Intine is the innermost layer, inner layer, and this is thin, and it is made up of cellulose and pectin. And this exine, it is discontinuous. It is not a continuous layer. It is having gaps in between, but intine is the continuous layer. Then pollen grain is having cytoplasm, and the cytoplasm of pollen grain, it is surrounded by plasma membrane. Then we learned about pollen allergy. Pollen grain, sometimes it may cause allergy. Some species plants will be having pollen grain which is causing allergy, especially anemophilous plants that cause several allergic and bronchial problems. especially Parthenium, that type of plants causes pollen allergy. Then Parthenium, Amaranthus and all we have learned as example. Then pollen products, we learned about pollen products also. So the pollen grains, they are rich source of nutrients. So athletes and all using pollen tablets, they are the rich source of nutrients. Then we learned about pollen viability. Means up to a period this pollen will be viable. So during this period fertilization should take place, and pollen viability, it depends upon the species. Then what and all we have learned in this chapter. Then we learned about the female part, and we learned about pollen grain, like it is having like pollen grain or microspore that divides mitotically into two cells, vegetative cell and generative cell. Vegetative cell is bigger and generative cell is smaller. Then we learned about pistil, Megasporangium, that is ovule, and embryo sac. We learned about all these things. So the gynoecium, that is a female reproductive part of a flower, and that consists of single pistil, means monocarpellary. If that is having more than one pistil, it is called as multicarpellary. Okay. Then part of pistil we learned stigma, style and ovary. It is having three parts: stigma, style and ovary. Then we learned about ovule, structure of a ovule. Which type of ovule we learned? The structure anatropous ovule, that is mostly seen in almost all the plants. So the structure of ovule can be studied mainly which and all the main parts for the ovule. Funicle. Funicle is the stalk of the ovule by which it is attaching to the placenta. Then hilum. Hilum is the junction between ovule and funicle. Then next comes the integument. Next is the integument. That means integument is one or more. That is a protective envelope which is surrounding the body of the ovule. Then micropyle, that is a pore or the passage which is present at the tip of the ovule, where that integument is absent. In that micropylar region, integument will be absent. That is an opening. Then chalasa, opposite to the micropylar region, that is the basal portion of ovule. Okay. So nucellus. Nucellus is nothing but the parenchymatous mass of tissues which is enclosed within the integuments, and that forms the body of ovule. Nucellus forms the body of ovule, and depending upon the development of nucellus, ovules are of two types. Then here starts the classification of ovule. This is depending upon the nucellus. Ovules are two types: crassinucleate ovule. In this, nucellus is well developed in crassinucleate ovule, and tenunucleate ovule. Okay, tenunucleate ovule, crassinucleate ovule and tenunucleate ovule. In tenunucleate ovule, the nucellus is poorly developed. It's not well developed. So these are the classification based on nucellus. Next, we learned in an ovule embryo sac. This is also called as female gametophyte, and it is located in the nucellus. So an ovule generally it is having single embryo sac formed from a megaspore. And the types of ovule we learned on the basis of relative position of micropyle, body of the ovule and funicle. So it depends. This classification mainly depends upon the degree of curvature, and there are 6 types of ovules like that: orthotropous ovule, anatropous ovule, hemianatropous ovule, then campylotropous ovule, then amphitropous ovule and sessinotropous ovule. So there are different types of ovule based on the micropyle position, then body of the ovule and the funicle. Okay, so in orthotropous ovule, micropyle, chalaza and funicle are in a straight line, but in anatropous ovule, the ovule turns at 180 degree. In an anatropous ovule, there is a complete 180 degree angle, it will rotate. And hemi-anatropous ovule, the ovule turns at 90 degree angle upon the funicle. Okay, then compylotropous ovule means that ovule is curved more or less at right angle to the funicle. Okay, the ovule is curved and it is right angle to the funicle. Then amphitropous, in this ovule and the embryo sac is curved like a horseshoe, it is curved. Okay, then succinotropous ovule means that ovule turns more than 360 degree angle. So this is the classification based on relative position of micropyle, then body of ovule and funicle. Okay, it's based on the degree of curvature. Then we have learned different topics of the sexual reproduction of flowering plants. So next we are going to learn about development of embryo sac or female gametophyte. Okay, so just before learning about the female gametophyte, we learned about the megasporogenesis. Okay, megasporogenesis means it is the process of formation of megaspore from megaspore mother cell, that is called as megasporogenesis. And in this, ovules generally differentiate a single megaspore mother cell or MMC in micropylar region of the nucellus, right? A single megaspore in the micropylar region of nucellus, and this megaspore mother cell, that is a large cell. It is having dense cytoplasm and a prominent nucleus. Okay, then this megaspore mother cell that will undergo meiosis and it forms 4 linear tetrads. And out of this, one will be functional and other 3 degenerates. Okay, so the step is development of embryo sac or female gametophyte. So what do you mean by gametophyte? Gamete bearing body, right? Phyta, that word is related to plant body or the body. So gametophyte means gamete bearing plant body, isn't it? So female gamete bearing body, that is called as female gametophyte. Now the megaspore, it is in linear tetrad form. Here you can see the linear tetrad form of a megaspore, right? That is after meiosis. So it is in tetrad form. So in most of the flowering plant, among these 4 megaspore tetrad, 3 degenerates and only 1 exists. Okay, so the one towards the chalazel end that exists. That existing megaspore, that will become functional. Okay, so which is functional, that existing, which is towards the chalazel end, that is existing and that will become functional, and only that will be functional. Other 3 will degenerate. Okay, other 3 will degenerate. Micropylar side cells will degenerate. Okay, so that existing one is called the functional megaspore, the only one which exists, that is the functional megaspore. So that functional megaspore that develops the female gametophyte or the embryo sac. Okay, this develops the female gametophyte or embryo sac. So in most of the angiosperms, chalazel megaspore remains functional. Okay, in the case of most of the angiosperms, the chalazel one will become functional, and this method of embryo sac formation from a single megaspore is called as monosporic development. Okay, so what is that? What are all the things happening here? female gamete bearing body, that is the female gametophyte. The megaspore, here you can see after meiosis it is in the tetrad formation, or it is in the tetrad form. So in most of the flowering plant, among these tetrad, 3 degenerates and only 1 exists, right? So the one which is that one which is towards the chalazal end, here you can see up the 3 degenerates and the down one is existing, which is towards the chalazal end, that is existing, and the one all the 3 other cells which is towards the micropylar region, that degenerates. Okay, so those cells degenerates, and the chalazal which is towards the chalazal end, that is the one which is existing. Okay, so that is called as functional megaspore, which is existing. The chalazal side one is existing, that is called as a functional megaspore. So that functional megaspore that will develop into female gametophyte. Here you can see when it is going to next stage, this is becoming enlarged and the other 3 that degenerates and it is going to disappear. So this functional megaspore develops into a female gametophyte, or this becomes the embryo sac. Okay, so in most of the angiosperms, which one is the functional megaspore? The cell which is towards the chalazal end, that becomes the functional megaspore. And such a method of embryo sac formation from a single megaspore, that is called as monosporic development. What is monosporic development? The development of embryo sac, or the embryo sac formation from a single megaspore, that is called as, here only this is the functional megaspore. From that functional megaspore, embryo sac formation is taking place, that is called as monosporic development. Then we'll see the ploidy of cells. So you have to note down this point because it is very important. The ploidy, here most of the questions comes with this ploidy. So nucellus, we said the parenchymatous cells of the nucellus. So it is in diploid condition, or it is having 2 sets of chromosome. It's in 2n condition, right? Then the microspore mother cell, that MMC, it is in 2n or diploid condition. Because of that, it is undergoing meiosis, right? The microspore mother cell is having 2 sets of chromosome, or it is a diploid cell, so it is undergoing meiosis. Then what is the ploidy of functional megaspore? You can tell the ploidy of a functional megaspore. So you know it's in a haploid condition because the functional megaspore, it is formed after meiosis. Here you can see it is in tetrad formation, and all these 3 degenerates and the last one which is towards the chalazal end, that exists. So that becomes the functional megaspore. So what will be the ploidy of this cell after meiosis? After meiosis, it is the haploid cell, right? It is having only 1 set or n number of chromosome. Okay, so the functional megaspore is forming after which one? After which cell division? After meiosis. So after meiosis, a functional megaspore is forming. Here it is in tetrad formation, and the 3 cells degenerates, and the one which is existing, that is also a product of meiosis. So definitely the ploidy of that cell will be haploid, or it is having 1 set or n number of chromosomes. Okay. So here in a majority of flowering plants, one of the megaspore is functional while the other 3 degenerates. So only the functional megaspore develops into female gametophyte or the embryo sac. And this method of embryo sac formation from a single megaspore, that is called as monosporic development. That is called as monosporic development. Clear? And next is, I said megaspore tetrad, that 3 cells degenerates and 1 is becoming a functional megaspore, right? So now let's see the further developments which is taking place inside the functional megaspore for the development of embryo sac, right? We said all the 3 cells of that tetrad that degenerates, and the functional megaspore which is towards the cell and is existing. And next, the further developments for the development of an embryo sac. Okay, let's see each and every step in this. So in the starting stage, our functional megaspore is a single nucleate stage. Here in the previous picture you can see it's a single nucleate stage in the starting stage. So next, that means it is a cell with a cytoplasm, right? There, after the degeneration of other three cells, this cell is a single cell which is having a nucleus and cytoplasm, and it is having single nucleus, right? So in the next step, what happens? In the next step, the nucleus of this functional megaspore divides mitotically. So here meiotic division up to cell formation, 4 cell tetrad formation, it's having that meiotic division. So next comes mitotic division. Okay, when that 3 cells are degenerating, and after that 1 cell which is towards the cell end is existing, then next division for this functional megaspore, that is it's not a meiotic division, it's a mitotic division. So which division? Mitotic division is taking place. So in the next step, the nucleus of this functional megaspore, note down, nucleus of the functional megaspore divides mitotically, and that forms 2 nuclei. Here you can see 2 nuclei. Here nucleus of that functional megaspore is dividing and it is forming 2 nuclei. Then these both nuclei move to opposite sides. Here you can see it is moving towards opposite side or opposite poles. Then it will be like embryo sac with 2 nuclei, right? Now this is like embryo sac with 2 nuclei. So the functional megaspore nucleus is getting divided into 2 and it is moving towards opposite poles. This is like embryo sac with 2 nuclei. Then each of this nuclei, which is, see already it is moving towards both poles, and that divided nucleus that divides, that means each nuclei will give rise to 2 nuclei, right? This is the, from this pole it is getting divided and it is forming 2 nuclei, and from that downward pole, that also it is dividing and forming another 2 nuclei, and it forms total of 4 nuclei in the next stage. Each nuclei which moved towards the opposite pole, that again it will get divided and it will form a 4 nucleate structure, okay, or 4 nucleate stage. Then there one more mitotic division for the nucleus, it is taking place, and to complete the sequence of division, and after this division there will be 8 nucleate stage in that cell. Okay, so one more mitotic nuclear division is taking place to complete the sequence of that division, and after that division there will be 8 nucleate stage in the cell. Okay, here you can see 8 nucleate stage. Okay, now that embryo sac is in 8 nucleate stage. And the very interesting and important thing is, all these mitotic divisions are only nuclear divisions. Okay, note down, that is only for nucleus, or free nuclear divisions are taking place. That means nuclear divisions are not following immediately by cell wall formation. This is strictly nuclear divisions. Usually how one cell is dividing, first nuclear division is taking place and it is followed by cytoplasmic division also, right? So karyokinesis is followed by cytokinesis also in general cases. Cytokinesis means that's nothing but the division of cytoplasm, and how that cytoplasm will get divided? By the formation of cell wall. So here, that's how generally one cell, karyokinesis, which is followed by cytokinesis, and after cell wall formation both cells will get separated. That is a general procedure. Okay, but here in the formation of embryo sac, all these divisions are only for nuclei. Only nuclear divisions are taking place. There is no cytoplasmic division. Now in each pole, Okay, here I haven't mentioned any cytoplasmic division, only nuclear division you can see here. So there are no cytoplasmic division. Now in each pole you can see 4 nuclei, right? In each pole, here in this pole also 4 nuclei are there, and this downward pole also you can see 4 nuclei, right? Out of 4, 1 nuclei from each end, and that means 1 nuclei from the micropolar region and 1 nuclei from the chalazel region, both migrates towards sender. Okay, here also you can see 4 nuclei, and downward this side also you can see 4 nuclei. Among this, from each side 1 nuclei will move towards the sender. That means 1 from the chalazel end and 1 from the micropolar region, both will move towards the sender. So at the center of embryo sac, 2 nuclei are present. Okay, here you can see, if this is here, this is the embryo sac, and from micropolar region 1 nuclei is coming, and from the chalazel region 1 more nuclei is coming. So at the, here you can see this is chalazel and 1 and micropolar end, and from both ends 1 nuclei are coming towards the center, and at the center there will be 2 nuclei. So these nuclei, they are called as polar nuclei. Here we have marked as polar nuclei because from both poles it is coming towards the center. So they are called as polar nuclei, and both polar nuclei are present at the center. Okay, polar nuclei from both poles, each nuclei, 1 nuclei are coming, and it is present at the center. So from both poles it came. It is called as polar nuclei. Okay. So here what and all we have told now, that functional megaspore, which is in the single nucleate stage, that will get divided, and the nucleus of that functional megaspore will get divided, and it is forming in the first stage it will be 2 nucleate stage, and again 1 more nuclear division is taking place, which is making 4 nucleate stage in that cell, and again it will undergo 1 more mitotic division to form 8 nucleate stage. To complete, there are 3 continuous nuclear divisions, and finally 8 nucleate cell will be formed, and that is the sequence of that division. And here you have to note down the main point is all these divisions are strictly nuclear, and there is no cytoplasmic division or cytokinesis is taking place. It's only karyokinesis, and that is not followed by cytokinesis. And from at each pole now you can see 4 nuclei, and from each poles 1 nuclei will move towards the center. So from each pole that nuclei, each one nuclei are moving towards the center. From the micropolar end 1 nuclei will move towards the center, and from the chalazel end another nuclei will move to the center. So at the center 2 nuclei from both poles will be there. So these are called as polar nuclei. Okay. So the nucleus of the functional megaspore divides mitotically, and that forms 2 nuclei which move to the opposite poles, forming 2 nucleate embryo sac. And 2 more sequential mitotic nuclear divisions result in the formation of 4 nucleate and later 8 nucleate stage of that embryo sac. So these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed by the cell wall formation. Okay, then after this 8 nucleate stage, that cell walls are laid down properly, and this leads to the organization of a typical female gametophyte or a typical embryo sac. Got it? So after the 8 nucleate stage, the cell walls are formed. Okay, you can see the cell wall formation, and that forms the organization of typical female gametophyte or the embryo sac. So now here, look at this diagram. How many nuclei are there? Here, how many nuclei are there? Total of 8 nuclei, right? So among these 8 nuclei here, 1, 2, 3, 4, 5, 6, 7, 8. You can see 8 nuclei in this. And among these 8, 6 are surrounded by new cell walls and formed into cells, right? Here you can see 1, 2, 3, 4, 5, 6. All these 6 here, again cell wall formation, 1, 2, 3, 4, 5, 6. Okay, 6 are separated with cell walls, right? So after 8 nucleate stage, that cell walls are laid down, leading to the organization of a typical female gametophyte. This is a typical female gametophyte or embryo sac, and 6 of the 8 nuclei are surrounded by cell walls and organized into cells. Here these 3, these are separated by cell wall, okay, and here these 3 also separated by cell wall, and the remaining 2 nuclei, they are called as polar nuclei, and they are situated below the egg apparatus in the large central cell. Okay, here this side 3 cells, this side 3 cells, and large central cell is present. In that 2 nuclei, polar nuclei are present. Okay. Then in this micropylar region, you can see 3 cells, right? Micropylar region, 3 cells, 1, 2 and 3 cells, and all these 3 nuclei present at the micropylar region formed into cell by cell wall formation. Here you can see all these nuclei are separated by cell wall formation. So in the same way, 3 nuclei towards the chelazal end. Here you can see the 3 nuclei which are towards the chelazal end. This is the micropylar end and this is chelazal end. So towards chelazal end also 3 nuclei are present, and those are also separated by cell wall formation, right? So in the micropylar end, you can see 3 now, right? 3 cells in the micropylar end, and among these 3 cells, 1 cell is large. 1 cell you can see that is larger than these 2 cells, and that is a special cell that is called as egg cell. This large cell, and it is a distinct type which is called as egg cell. This is the egg. Okay, and the remaining 2 cells which are similar, they are known as synergids. Okay, these are called as synergids, which are similar. Okay, these remaining 2 smaller cells, these are known as synergids, and these are similar, almost similar and small cells. And this large egg cell and 2 smaller synergids are collectively called as egg apparatus. Okay, what is egg apparatus? These 2 smaller synergids along with the large egg cell, that forms synergids. Okay, sorry, that forms the egg apparatus. So the synergids along with the egg cell, those are called as egg apparatus. And this egg apparatus, it is present at the micropylar region. Egg apparatus is present at the micropylar region. What is the reason for that, or why? That pollen grain germination and pollen tube formation. When a pollen grain is reaching on the stigmatic surface, that will start germinating, and that pollen, it will form a pollen tube, and through that pollen tube which is coming through that gynoecium, that is reaching to the micropylar region, right? So for the fusion of egg and sperm, that should be near the egg apparatus should be near the micropylar region, because through micropyle this pollen tube is entering inside, right? So one egg plus here one egg plus two cyanogates. These are called as egg apparatus. Then coming to the next end or the next pole, the chelacell end, 3 cells. Here you can see again 3 cells. This is the chelacell end, and here you can see 3 cells which are called as antipodal cells. Okay, antipodal cells. So in the large central cell, here this is the large central cell, this one. Okay, this is the large central cell, and at the center of this cell you can see the polar nuclei. Okay, polar nuclei are present in the large central cell, right? So there are 7 cells now, 1 2 3. These are 3 cells, and in the opposite micropolar region 1 2 3, 2 cyanogates and 1 egg cell in the micropolar region, and the chelacell and 3 antipodals. Okay, so now then the middle one is the large cell, the big cell which is having polar nuclei. So there are 7 cells now, right? This bigger cell 1 and 3 here, 3 and the chelacell end. So in total there are 7 cells and 8 nuclei. Why there 8 nuclei? Because in the middle cell or the large cell, in the central cell which is large, it is having polar nuclei, 2 polar nuclei, right? So there are 7 cells and 8 nuclei, right? So polar nuclei are present below the egg apparatus. Here this is the egg apparatus, and below the egg apparatus polar nuclei are present. Okay, so the polar nuclei are present at the central cell. So in the starting stage it will be like 2 nuclei. Okay, in the starting stage this polar nuclei will be separate only. Okay, then just before fertilization these 2 nuclei fuse to form a secondary nucleus or a definitive nucleus. So just before fertilization these 2 nuclei, they will fuse together in the center to form a secondary nucleus. Okay, and this secondary nucleus is diploid, right? Because 2 nuclei are fusing to form a secondary nucleus, and that is diploid in nature. So how many secondary nuclei will be there? Only 1 secondary nuclei. Okay, don't get confused. There will be 2 polar nuclei, and these 2 polar nuclei are fusing together to form a single secondary nucleus. Both polar nuclei, after fusion it is called the secondary nucleus. So both cells are fusing, so it is diploid. 2 cells are fusing which are haploid, and it is forming a diploid nucleus. So after fusion it's becoming 1. So there is only 1 secondary nucleus. Okay, 1 secondary nucleus which is diploid. Got it? So in an embryo sac there are the 3 cells are grouped together at the micropilar end. Here this is the micropilar end, and that consists of egg apparatus, or that forms the egg apparatus, which means cyanogates, 2 cyanogates plus which one? The egg cell, that forms the egg apparatus, and the cyanogates have a special cellular thickening at the micropilar tip that are called as filiform apparatus, that plays an important role in guiding the pollen tube into the cyanogates. So what are cyanogates? The cyanogates, they are finger-like structures. It's a kind of special cellular thickening at the micropilar tip. Okay, these are called as filiform apparatus, and with the help of these filiform apparatus or these structures, the cyanogates absorb food from the nucellus, that is one function, and that transfer into the embryo sac. Okay, and another thing, this guides the pollen tube towards the egg cell, and this filiform apparatus, they'll secrete some special kind of chemicals. This can attract and guide the pollen tube towards the egg cell. So that's a function of filiform apparatus. Okay, so the cyanogates have special cellular thickening at the micropilar tip. These are called as filiform apparatus, and this plays an important role in guiding the pollen tube into the synagogues. Okay, and three cells at the chalazal end also you can see three cells, and these are called as antipodals. Okay, those are called as antipodals, and the large central cell, here you can see a large central cell which is having two polar nuclei, and at the time of fertilization both polar nuclei fuse together to form a secondary nucleus. Okay, the large central cell which has two polar nuclei, and a typical angiosperm embryo sac in the at maturity, you can see there are 8 nuclei and it is in a 7 cell stage. Okay, a mature embryo sac will be having 7 cells and 8 nuclei. Why it is 8 nuclei? Because at the center the large cell will be having polar nuclei, 2 polar nuclei. Okay, and here next comes the details of the formation of this female gametophyte. Overall procedure I'll explain. So after 3 mitosis in the megaspore, the 7-celled and 8 nucleate structure is formed. Okay, so this 8 nucleate structure, this one, that 8 nucleate structure, it is called as female gametophyte. This is a stage by stage division. Here the tetrad formation, from that a functional megaspore is forming, all other 3 cells degenerates. Then first mitosis, that is 2 cell stage, then it forms the 2 cell stage, and second one again not 2 cell, 2 nucleate stage, and next in the next mitosis it forms 4 nucleate stage. There is only nuclear division, there is no cytoplasmic division is taking place, and again next mitosis that causes 8 nucleate stage, not 8 cells, 8 nucleate stage. Okay, so these 8 nucleate and 7 cell, only 7 cells are there but 8 nuclei are there. This forms a female gametophyte or the embryo sac of angiosperms. So this type of embryo sac is known as polygonum type. That type of embryo sac is called as polygonum type because it was discovered by Strasburger in Polygonum plant. Okay, this type of 8 nucleate and 7 celled embryo sac, mature embryo sac is discovered. It was discovered by Strasburger in Polygonum plant. So this type of embryo sac is called as polygonum type. So this is a common type of embryo sac which is present in angiosperms, and this polygonum type of embryo sac, it develops from a single megaspore. So it is known as monosporic embryo sac. Polygonum type of embryo sac is developing from a single megaspore. Here it is developing from a single functional megaspore. So it is called as monosporic embryo sac. Okay, so we have learned about monosporic embryo sac because it is starting from a single functional megaspore. So it is called as monosporic embryo sac. Okay, and it was discovered by Strasburger in polygonum type. That's why it is called as polygonum type of embryo sac. Okay, so hope all of you got the topic which we have discussed. Hope all are clear with this topic. Thank you.